Okay, so this is the last part of chapter 1. We were talking about the different types of receptors, and we talk about the GPCR and the TKR, which is the tyrosine kinase receptor. Today, we will talk about more receptors and of different types. The third type of receptor that we would like to talk about is the TKCR, which is the tyrosine kinase coupled receptor, or also called cytokine receptors. So please keep in mind that we have the classical nomenclature of the receptor, which is GPCR, TKR, TKCR, for example. And also sometimes we have next to it another common name, which describes a little bit the type of ligands that are going to be binding to the receptor. Like in this case here, we have cytokine receptors, but this is not exclusive. So keep in mind that the common name is not exclusive. So for example, when we talk in the second type of receptor about the kinase receptor, the typical ligand would be the insulin, but the receptor is also called a growth hormone, growth factors receptor. And this is why you should not be confused, like I just did, with the growth hormone, which is a ligand for the third type of receptor, the tyrosine kinase coupled receptor. Okay, so first receptor, GPCR, second one, tyrosine kinase receptor or growth factor receptor, third one, tyrosine kinase coupled receptor or cytokine receptor. The typical here that we will take is growth hormone or prolactin. So the ligands of these receptors are mainly cytokines and certain growth factors or hormones, such as GH, growth hormone, and the prolactin, or also leptin. So keep in mind always that the growth hormone and the prolactin, these two are going to be produced from the anterior pituitary, and they are going to be ligands to tyrosine kinase coupled receptors. These receptors are devoid of enzymatic activity. This is what makes them different from the previous ones, which are the tyrosine kinase receptors. When I say coupled receptors, that this means that they are going to be coupled to a kinase and not having the kinase activity themselves, okay? So these receptors are devoid of enzymatic activity. There is no enzymatic activity intrinsic to the molecule of the receptor itself in this protein, but it's going to be coupled to this kinase. Their di or trimerization induced by ligand binding results in the recruitment to the membrane and the activation of cytosolic tyrosine kinases. So it's a very important term here to understand that we are going to recruit cytosolic tyrosine kinases. So that will relay the signal downstream. So again, the tyrosine kinase coupled receptors, as their name suggests, is these receptors are going to have a tyrosine kinase activity, but it's not intrinsic to the receptor. Where does it come from? It comes from a cytosolic tyrosine kinase. So they are going to be coupled to a tyrosine kinase. Of course, tyrosine kinase is always a kinase that will, so it's an enzyme that will phosphorylate tyrosine residues. Okay. And for regarding the dimerization or trimerization, this is also classical. We already saw in the second type of receptors, the tyrosine kinase receptors, that they also going to dimerize, like the insulin receptor. We said that this is an exception. It's already dimerized when the ligand binds, but other receptors such as the epidermal growth factor, the EGF, when it binds, it's going to recruit the other part of the receptor, the second dimer, and in that case, it was a homodimer. Here we will see that we have heterodimerization, so the two subunits are not going to be completely identical. So, but the thing that is classical is that once I have a ligand coupled to the receptor, this will induce dimerization, and the activity of the receptor by relaying the signal downstream. We can distinguish tyrosine kinases of the JAK family that are associated with the cytokine receptors and are devoid of SH2 or SH3 domains, or tyrosine kinases of the SRC family. So we have tyrosine kinases of two different families that are possible to be coupled with these receptors. Either you can talk about the JAK family or about the SRC family of kinases. The examples that we have here is with the JAK family, Janus-like kinases. Okay, and they are associated with the receptors, and they do not have neither SH2 or SH3 domains. Remember that these domains are domains of interaction between with different proteins, and between the proteins. SH2 is the domain for phosphotyrosines, and SH3 is for proline-rich sequences. The main signaling pathway of the cytokine receptors may be described as follows. Should I go through this before the figure? Yeah, let me start with a paragraph and then we go to the figure, okay? So keep in mind the different steps that I will talk about now. The main signaling pathway of the cytokine receptors may be described as follows. The binding of the cytokine causes heterodimerization and transconformation that allows recruitment to the membrane and activation of the associated JAK tyrosine kinase. So first step is the binding of the ligand that will cause heterodimerization of the receptor, transconformation, with this heterodimerization is going to be associated with transconformation that allows recruitment to the membrane of the kinase, which is the JAK tyrosine kinase. Okay, recruitment and its activation. JAK, when it's going to be recruited, it's going to transphosphorylate. So each one of the kinase is going to activate the next one next to it. After this step, I have another phosphorylation step that corresponds to the phosphorylation of the receptor itself. Of course, always on tyrosine residues, creating anchor sites, binding sites, for cytosolic substrates, proteins with SH2 domain. In particular, the transcription factors of the STAT family. So هيدا دايما بس بدي احكي عن التيروزين kinase coupled receptor. I have always to keep in mind that an example that I'm gonna give, it should contain JAK-STAT signaling pathway. JAK-STAT, JAK بأول part, and then that will recruit STAT. JAK then will phosphorylate STAT, causing their detachment of the receptor and their dimerization. The dimer of STAT is going, in fact, to translocate to the nucleus and play the role of a transcription factor by binding to specific DNA sequences and activate a series of target genes. So in this figure, this is a figure that shows you the different steps. So here you have, one receptor that is going to be associated with this tyrosine kinase TYK2, and here you have another receptor that is associated with the tyrosine kinase JAK. Good, very good. Now I do not have a ligand. Once the ligand is bound to one of these two receptors, it will recruit the other one to approach. Okay, and once I have this conformation, the JAK and TYK2 are going to be recruited to the membrane, next to the membrane, and they are next to the receptor. They are non-covalently attached to the receptor. In this form, they are going to transphosphorylate each other. So JAK phosphorylates, for example, TYK2, it becomes phosphorylated, and TYK2 phosphorylates JAK. After this transphosphorylation of the enzyme itself, then the enzyme will start to phosphorylate tyrosine residues on the receptor. So لحديت هلا already I have two steps of phosphorylation. Okay, activation يعني. So binding of the ligand, heterodimerization, and then activation of JAK and TYK, which are associated with the receptors or coupled to the receptors. They will transphosphorylate each other, and after this, they will phosphorylate the receptor on tyrosine residues. All these phosphorylations are taking place on tyrosine residues. Okay, so let's go back to the previous page. Then the binding of the cytokine causes heterodimerization and transconformation that allows recruitment to the associated JAK tyrosine kinases. Very good. JAK transphosphorilates يعني JAK عملت phosphorylation لتيك وتيك عملت phosphorylation لJAK and then phosphorylates the receptor. Okay, creating anchor sites for cytosolic substrate protein that have now the SH2 domain, which are the STAT family. Let me show you the STAT. Now, by creating these phosphotyrosine on the receptor itself, the JAKs, they created anchoring sites, يعني binding sites to some cytosolic protein that have SH2 domain, يعني that would recognize phosphotyrosine, okay, specific phosphotyrosine, and then are called STATs. So as you can see, for example, you have STAT1 here and STAT2 that are going to recognize هيدا الphosphotyrosine and to bind to it. Like أنا هون I created docking points to the STATs family of proteins that now are going to be recruited to the level of the receptor, and next step is to phosphorylate the STATs on the tyrosine residues. They are going to be phosphorylated themselves. Third step of phosphorylation. Okay, after the phosphorylation of the STATs, they are going to detach from the receptor and to dimerize between each other through their SH2 domains and to become a complex that is going to be an active complex as a transcription factor. Translocate as transcription factor to the nucleus. Okay, so let's go back to read the last two steps. JAK then phosphorylates STAT. من بعد ما STAT قعدت على التيروسين residue through its SH2 domain, JAK will phosphorylate STAT, and then after its phosphorylation, this will cause its detachment from the receptor and the dimerization. Of course, هون عم نحكي عن STAT. The dimers are going to be translocated to the nucleus, where they bind specific DNA sequences and activate a series of target genes. Okay, so this form here, this complex here, is going to be formed by two STATs phosphorylated and attached to each other. This complex is considered as transcription factor, and then it will relay the message to the nucleus, where it's going to bind to specific sequences and activate their transcription. This slide, I like it, and I would recommend you to study from this slide, لأنه it will in fact make it much easier for you, because you have the figure and the explanation next to it. So the different steps are written here with all the details. So you consider you have alpha interferon as a ligand for this tyrosine kinase coupled receptor. You can consider that the tyrosine kinases are associated to the receptor or coupled to it, like in the same way the G protein is coupled to the G protein coupled receptor, right? To the GPCR. So they are here, but they are not active. They are not going to be activated unless I have a ligand. So binding of interferon cross-links adjacent receptors. It will cross-link the adjacent receptors, and the JAKs cross-phosphorylate each other on tyrosine. So بس الكلمة اللي مش مكتوبة هون واللي هي مهمة لإلنا هي ال changing of the conformation. لأنه when I have the alpha interferon, for example, ligand binding to the receptor, not only it's going to dimerize the receptor, this will create changes at the level of the tyrosine kinases that are going, in fact, to be activated. So, and JAKs cross-phosphorylate each other on tyrosine. So this JAK here will phosphorylate this one, and this also tyrosine kinase will phosphorylate this one on these tyrosine residues. The next step is that each one is going to phosphorylate the receptor. Activated JAK phosphorylate receptors on tyrosines. Now, by creating these phosphotyrosines here, I created docking points by recognition and binding points to specific proteins that are called STATs. Now, these proteins, they do have like this part here, is H2 domain. Okay, like it's written here, it's H2 domain. اللي هي light part of the STATs. The SH2 domain is a phosphotyrosine recognition and binding domain. So as you can see, they are going to bind through these domains to the phosphotyrosines on the receptor. So next, what will happen? That the third phosphorylation process will take place by the tyrosine kinases, of course, on the tyrosine residues on the STATs themselves. So I have 3 steps of phosphorylation now that STATs are phosphorylated, they are going to detach from the receptor. So after STATs dock on specific phosphotyrosines on the receptor, the JAKs phosphorylate them. And after this, the STATs dissociate from the receptor and dimerize via their SH2 domain. This is nicely represented to show you how the SH2 domain of each of the STATs is going to interact with the phosphotyrosine of the other one so that they are going to dimerize through their SH2 domains. ال SH2 domain لأول STAT for example بيعمل binding على الفوسفوتيروزين للثاني and vice versa. So you have this complex that is going to be formed. This complex STATs migrate to nucleus, bind to DNA like this. Of course مش حيلا DNA sequence. They are going to bind to, for example, if I consider إنه هون ligand is alpha interferon, they are going to target alpha interferon response element. يعني the genes that depend on the alpha interferon binding. وإلا شو هو ال specificity of this message, right? وإلا كيف بدي حافظ على specificity of the message يلي هو جاي من الإكسترا سيلولار للإنترا سيلولار message. Fine. So the interferon response element in target gene, and then they will lead to its transcription. So by this, the change that is taking place in the cell is change at the level of the transcription of genes, target gene transcription. Okay, so of course they can recruit other regulatory proteins, co-activators next to them in order to allow this transcription to take place. So this is about the third type of receptors, which was the tyrosine kinase coupled receptors or cytokine receptors. The fourth type of receptor that we would talk about is the serine threonine kinase receptors, or that we also call TGF-beta receptors for transforming growth factor beta. So also from the name here, you will all directly recognize that these receptors, they do have a kinase activity, and this kinase activity, since there's no coupled word here in the name, so then the kinase activity is intrinsic to the receptor itself that is playing the role of enzyme at the same time, but then you pay attention that the only thing different here is that they are going to be phosphorylating serine and threonine residues rather than tyrosine residues. Serine or threonine residues or amino acids. Good. So what are the characteristics of such receptor, serine threonine kinase receptor? Serine threonine kinase receptors have common structural and functional characteristics, all of them. And this is a family, of course, of receptors. What do they have? A single polypeptide chain composed of 3 regions. هلا لاحظوا انه كل الريسبتورز اللي حكينا عنهم من بعد ما حكينا عن الجي بي سي ار كلهم they are composed of one single polypeptide chain. ما بس يعني هلا كلهم هن single polypeptide chains بس one transmembrane passage قصدي انا one segment يلي هو بقلب الممبرين. while انه بالجي بي سي ار كان في عندنا 7 segments بتفوت وبتظهر 7 times the GPCR, where the others are just like single polypeptide chain composed of 3 regions. The N-terminal extracellular region, highly glycosylated, so it is a glycosylated protein, هيدا receptor, with its N-terminal terminus is going to be extracellular, and it is corresponds to the ligand binding domain. This is where the ligand is going to bind. Then it has a short transmembrane sequence that crosses the membrane, and an intracellular C-terminal region that provided with the serine threonine kinase activity. This region not only has the enzymatic activity, which is serine threonine activity, but also it has several phosphorylation sites of serine and threonine residues. So بتكون في عندها serine وthreonine residues able to be phosphorylated by its own activity. Okay, they also serine threonine kinase receptors, they also dimerize upon ligand binding. After the ligand binding, they are going also to dimerize, like previously in the two previous types of receptors that we talked about. كلهم they are going to dimerize. وهون في عندي كمان a heterodimerization. Okay, the two subunits are not completely identical. So if you take an example on the TGF-beta receptor, once I have TGF-beta bound as a ligand to the receptor, it will in fact recruit the other type of receptors. Let's talk about it first to define each subunit and give it a name. TGF-beta receptor is a heterodimer consisting of two subunits. One of type 1, so عندكم TBR1 which يعني TGF-beta binding receptor 1, and the other subunit هي ال TBR2 which correspond to TGF-beta binding receptor type 2. Okay, حنقول نحن في عنا TGF-beta receptor type 1 و TGF-beta receptor type 2. هو اثنين subunits هذول اللي بيألفوا الريسبتور تبع TGF-beta. But what is the sequence of events that is going to take place here? The ligand TGF-beta is going to bind to TBR2 first. Okay, it binds to the TGF-beta receptor type 2. And then it allows the recruitment of the other subunit, which is the R1. So بيعمل binding على R2, بيعمل recruitment لل R1, and then activation of the serine threonine kinase domain. Okay, activation يعني للenzymatic activity of the TBR2. TBR2 will phosphorylate TBR1, okay, and then the TBR1 will phosphorylate the transcription factors like previously كانوا stat. هون بدي أفكر دايما بالسماد. اوكي دونك بس احكي عن tyrosine kinase coupled receptor بدي احط ببالي انه this corresponds to the signaling pathway JAK-STAT. وبس احكي عن السيرين threonine tyrosine kinase serine threonine kinase receptors بدي احكي عن SMAD factors. It will phosphorylate the transcription factors SMAD, and then these SMAD2 and 3, once they are phosphorylated, they will dimerize with another SMAD called SMAD4, and then I have a heterodimer that corresponds like previously, like a transcription factor that is going to be translocated to the nucleus and change the transcription of certain genes. You will understand all this better when I show it to you on figures. So I have TGF-beta ligand, and I have two subunits of the receptor. A light subunit is TGF-beta receptor type 2, and the green يلي هي الفونسي here, okay, it's type 1 TGF-beta receptor. Good. So first thing taking place is the ligand binding to the TGF-beta type 2 receptor. And then I will have the association with the other type 1 TGF-beta receptor. Then what will happen is the enzymatic activity, the serine threonine kinase domain of the TGF-beta receptor type 2 will phosphorylate the type 1. And now the type 1 will further phosphorylate the SMADs, okay? Here I will have phosphorylation of the type 1, and then the type 1 will in fact phosphorylate the SMADs. And then the SMAD detach, they are phosphorylated as well. The SMAD2 or SMAD3, once they are phosphorylated, they dissociate, they bind with SMAD4, and they form the complex that is going to translocate to the nucleus and now is considered as an active transcription factor, as you can see, that will recruit other co-activators and allow the transcription. Also, for this type of receptor, I have a nice slide that shows you the different steps with the figure and the explanation next to it. So also I recommend to you to study from this slide the type of activity of this receptor. So TGF-beta binding of type 2 receptor causes the receptor to recruit and phosphorylate the type 1 receptor. Phosphorylated type 1, phosphorylated on serine threonine residues. Phosphorylated type 1 receptor recruits and phosphorylates SMAD2 or SMAD3. So this protein here is called SMAD2 or can also be SMAD3. Whatever, whatever protein from these two is going to be recruited to the receptor and it's going to be phosphorylated. By the receptor, okay, by R1. Now, once it is phosphorylated, it's going to detach and it's going to oligomerize with SMAD4. يعني another protein here that no need for it to be phosphorylated, this one here. So unlike the stats previously, SMAD دايما بده يكون الهيترودايمرايزيشن هي between SMAD2 مع SMAD4 أو SMAD3 مع SMAD4. والقصة إنه لازم يكون SMAD2 أو SMAD3 phosphorylated. Okay, SMAD4 no need for its phosphorylation. So phosphorylated SMAD2 or 3 dissociate from the receptor and oligomerize with the SMAD4. Now this is an active complex. The SMAD2 or 3 with the SMAD4 oligomer migrates into the nucleus, recruits other gene regulatory proteins, and activate transcription of specific target genes as previously. These consensus sequences where, or what we call response elements, are specific. And in this case here, since the ligand is TGF-beta, they are going to be called TGF-beta response elements in the target genes. لكن هون بده يروح يتعرف على TGF-beta response elements, بينما بالprevious type of receptor حكينا عن alpha interferon response element that are going to be recognized by the transcription factor يلي كان STAT1 and STAT2. So keep in mind that these elements, they are always specific and that they depend on the ligand that is going to bind to the receptor. So في عندي أنا target لهيدا اللايجند محدد يلي هو بده يجي الترانسكريبشن فاكتور بآخرة المطاف تبع السيجنالينج باثواي يعمل له activation أو transcription. Okay. So this is about the serine threonine kinase receptor يلي هو was the fourth type of receptor, serine threonine kinase receptor or TGF-beta receptor. Okay. So did you see the difference between this type and the previous type? Okay. دايما بدي اتذكر انه بالتالي التايب بدي احكي جاك ستات، بينما بس احكي عن سيرين تريونين كاينيز ريسبتورز بدي احكي عن السماد. Okay, and this will make it easier for you. Any questions for now? Okay, so last part that I would like to talk about is about the nuclear receptors. The nuclear receptors, I will talk previously today about the nuclear receptors. We will have the occasion to talk a little bit more about them in the chapter 3 when we talk about the thyroid hormones, because the thyroid hormones, they do have nuclear receptors. So I have a first example here for the nuclear receptors. أول شيء nuclear receptors هن عبارة عن proteins موجودين either in the nucleus or in the cytoplasm, and they are not at the surface of the cell, which makes the big difference between the other receptors, which are surface receptors. And usually they are present and they are going to be activated once the ligand is present inside of the cell as well. Okay, so once the ligand is going to be inside of the cell, these receptors are going to be activated and they will play their role. So the pathways here are completely different, لأنه ما ضروري إنه يكون في عندي شيء بده يوصل بين السيرفيس لعند النيوكليوس for example. لا، لأنه هون الريسبتور itself is not at the surface of the membrane. So let's take the example that we have on this slide, which is the estrogen signaling classical genomic pathway. This is a classical genomic pathway. Okay, so I have, for example, estrogen receptor alpha, ER alpha, estrogen receptor alpha, and I have the E2. As you can see, if I do not have the ligand, the estrogen receptor alpha is going to be sequestrated in the nucleus through a protein called HSP that keeps it like inactivated. Once I have E2, which is the estrogen ligand, so this is a steroid hormone produced from the cholesterol, as if you remember, through steroidogenesis in some specific tissues such as the gonads. Okay, estrogen E2 is going to enter the cell since it's hydrophobic. So it has the ability to enter the cell and to enter to the nucleus. When I have the estrogen E2, it will in fact replace the HSP and bind to the ER alpha. E2 with 2 ER alpha, يعني a 4 subunits complex here, is going to is going to be considered as an active transcription factor and recruit co-activators that will allow the transcription of specific genes that have, in fact, estrogen response element, ERE, estrogen response element, okay, which are present on targeted genes and that will be, in fact, transcribed. Examples of estrogen targeted genes in the cyclin D1 and CEMIC. These two genes are important in the cell proliferation and in the cell growth, and in fact, these are targets of the estrogen in the tissue where estrogen is going to be active. Okay, so these two types of genes, they do have estrogen receptor element in their sequence, and this is what will allow them to be, in fact, activated once the estrogen is going to be increased in this cell. Okay, so what happened؟ برجع بعيد إنه في عندي ال ER alpha موجود بقلب السيل but it's not active لحاله. ال ER alpha is not an active transcription factor. It's not going to be it's not binding to the estrogen response element. لحتى يصير في عندي activation لازم يكون في عندي E2 bound to ER alpha و2 it's a tetramer هون منه مبين لأنه I'm working with a PDF not with the PowerPoint, but it should be animated this. Okay, but I will show it to you on the next slide. Maybe it's better. So if you can see on the next slide here, I have the the estrogen receptor, and I have the estrogen. The estrogen will bind to the receptor, steroid binding. And when I do have this binding, أول شيء حيصير في عندي estrogen receptor will be separated أو dissociated from the HSP يلي قلنا عنها is bound to it. And then once I have this binding, I have conformational change and I have dimerization. I need another also pair of estrogen receptor that will allow the dimerization. Now I have my complex complete with 4 subunits, 2 estrogen with 2 estrogen receptors that is now active, that will bind to the DNA. At what level it will bind of the DNA? حيلا gene, of course, not on the genes that contain estrogen response element لأنه this is the pathway for the estrogen. طبعا أنا I will target the genes that respond to the estrogen and سميناهم نحن scientists يعني estrogen response element يلي هن they are going to bind to this complex that will recruit co-regulators and in particular co-activators that will recruit other proteins that you can see here. And in particular, it will recruit the RNA polymerase II complex, which is the enzyme that will allow the transcription of the gene in order to give me messenger RNA, and then the gene is going to be expressed. Okay, the target gene expression. Now, I do have يعني انا عم بحكي باختصار النيوكليار ريسبتورز بس هو بالحقيقه يعني النيوكليار ريسبتورز في عندي كمان نيوكليار ريسبتور 1 نيوكليار ريسبتور 2 as types يعني. Okay, so as I said previously, we will talk about them later on. Let's take another example that is a little bit different. As I already said, هيدا example we will talk about it more in details in the thyroid hormone chapter. In this case, when I talk about the thyroid hormones are كمان like the steroids, so they also have nuclear receptors and not a surface receptor. So they have the ability to cross the plasma membrane and to enter the cell and to reach their receptor. T3 is the active hormone of the thyroid hormones that enters the cell, and then it's going to bind to thyroid hormone receptor, THR, thyroid hormone receptor. بس بعدني ما صار عندي الكومبلكس that is going to be active. This ligand مع receptor are going to dimerize with another nuclear receptors called موجود in the majority of the cells of our organism. Retinoic acid type of nuclear receptors. So I have the formation of this heterodimer. And heterodimer is going also to be activated and is going to يعمل binding a thyroid response element. Here we call them thyroid response element. In the previous case we called them estrogen response element. So شو هي الهرمون بتزيدوا عليها response element؟ تيجي beta response element, thyroid response element, estrogen response element, testosterone response elements. All they are specific elements that are going to be responsive to the transcription factors activated through signaling pathway مع هيدا الهرمون بالتحديد. Okay, and then in this case is also going to be activated and they will allow the intermediary co-activated proteins to allow the transcription of certain targeted genes. Later on I explained this with much more details لأنه هون في عنا details مثل إنه حتى لو كان منه bind with the ligand, sometime I do have an activity for the receptor بقلب السيل before the binding of the ligand, ولكن هالactivity ممكن تكون معاكسة للactivity after binding of the ligand. So this we will talk about it later on. So نحن اللي بدنا نعرفه إنه أول شيء nuclear receptors are present in the cytoplasm or in the nucleus. They are transcription factor and inactive transcription factor لحد ما يكون في عندي ligand binding. بعد ما يصير في عندي ligand binding formation of the complexes, then they are going to be considered as transcription factors and they are going to be bound to specific elements called in general hormone response elements that will allow the recruitment of co-activators that will allow the transcription of the targeted genes corresponding to each hormone. now I have two slides just okay, before talking about these two slides, let me just talk about the last slide. Now, I am done with all this. I am done with the nuclear receptors. I will just show you the last slide of the chapter that correspond to the insulin receptor, which is a tyrosine kinase receptor, TKR. And the two main pathways that are going to be activated by insulin, like we will also see in the chapter corresponding to the pancreatic hormones. If you remember that I said that the insulin has a receptor, tyrosine kinase receptor, that is already dimerized, يعني already في عندي 2 dimer. And then it's going to be transphosphorylated, okay? Each subunit is going to phosphorylate the other subunit, creating, of course, tyrosine residues that are phosphotyrosines that will recruit specific يعني هيدا proteins through their specific domains. ممكن يكون SH2 domain، البعض منهم عنده SH3 domain مثل GRB2 كمان عندها SH3 or pH for plextrin homology domain that is going to recruit them to the membrane. So نحن لما نحكينا عن الانسولين قلنا انه في عندي two main pathways that are going to activate it. واحد سميناه الماب kinase pathway, which is this one here, MAP kinase pathway, and the other one سميناه AKT pathway, which is this one here. Okay, so these are two main pathways that are going to be activated by the insulin. وبآخر المطاف كل واحد منهم بيعمل لي activation ل different types of protein بقلب السل depending on what type of cell or target cell I have for the insulin. So the only thing here is that in this figure you see IRS and SHC, which is called the chi. So these two proteins, we didn't see them previously. So this is why I just introduced two slides in order for you to recognize these two proteins if you want. So IRS first is insulin receptor substrate, called insulin receptor substrate. IRS, of which there are 4 isoforms, IRS 1, 2, 3, or 4, of different tissue expression, has in its N-terminus end a pH domain whose role is to recruit the protein to the membrane by interacting with negatively charged phosphate groups of specific membrane phosphoinositides. So ال IRS عندها pH domain، وهيدا ال pH domain هو اللي بيخليها تعمل interaction مع الممبرين. So let's look على ال IRS. IRS is from the AKT pathway، وال AKT depends on the phosphoinositide بقلب الممبرين. Remember إنه هيدا ال pathway ببلش بال PI3 kinase which will phosphorylate ال PIP2 to PIP3، وبس يكتر عندي ال PIP3 بيعمل activation لل PDK1 و 2 that will in fact activate ال AKT. اوكي يلي هي ال PKB. ال AKT فينا نسميها PKB كمان نفس الشيء. So نحن عم نحكي عن البروتين IRS هون، عم نقول انه في عندها pH domain، عندها pH domain بيخليها تتقرب من الممبرين، يعني بيعمل interaction مع الممبرين. Then it has also a phosphotyrosine domain, a binding domain whose role is to interact with the phosphotyrosine of the juxtamembrane domain of the receptor. هون يعني it's going to interact with this through the PTB, and it has also many sites of tyrosine phosphorylation whose role is to interact with the SH2 domain of other protein such as PI3 kinase. ال SH2 domain تبع ال PI3 kinase يعني في عندها phosphotyrosine that are going to be phosphorylated. اوكي لكن ال IRS في عندها ال pH domain to interact with the membrane، عندها ال PTB domain to interact with the phosphotyrosine of the receptor، عندها tyrosines that are going to be phosphorylated by the receptor in order to interact with the SH2 domain of the PI3 kinase, so that I have all the activations like downstream taking place. اوكي رح اذكركم فيهم. لكن ILS recruited and phosphorylated that this will recruit ال PI3 kinase which will اتذكروا انه PI3 kinase كنت مؤلفه من اثنين subunits ال P85 وال P110. وحده منهم عم تعمل باي اتس SH2 domain عم تعمل interaction مع هودي الفوسفوتايروزين وبتعمل activation للكاتاليتيك activity يلي حتعمل لي من ال PIP2 PIP3 and I have enrichment in the membrane of PIP3 here which will induce in fact an activation of the PDK الفوسفاتيديد يعني dependent kinases. These kinases are going to phosphorylate ال AKT يلي هي PKB وال PKB هي آخر المطاف هي الكاينيز اللي أنا بدي إياها هون بآخر المطاف لحتى تعمل لي phosphorylation ل different types of protein such as glycogen synthase kinase وبس تعمل لي لها phosphorylation هيصير في عندي activation للglycogen synthesis and this is like in the case of hepatocytes إذا أنا بالliver I need to activate the glycogen synthesis after the insulin binding لأنه أنا I want to stop the glucose under the form of glycogen. So this is the pathway. Okay, more details will come in the chapter corresponding to the insulin. And now the other بالتاني pathway I also talked previously about the GRB2 SOS RAS RAF MEK MAP kinase وكل هيدول حكينا عنهم. I will just introduce to you the Chi, the SHC. It plays a similar role to that of IRS and has two binding domains to phosphotyrosine, PTB and SH2 but not to the PH domain. لكن في كمان عندها binding للphosphotyrosines الكاي. اوكي والجي ار بي 2 كمان عندها binding للphosphotyrosines تذكروا through SH2 domain رح تعمل binding مع الكاي وينعمل لها هي عندها SH3 domain حتعمل فيه recruitment للson of seminless يلي هو ال SOS ويلي هو حيعمل activation للراس. الراس is a small G protein يعني هون بصير في عندي exchange للجي DP by GTP activation للراس و finally activation لكل هيدي sequence of الماب kinases يلي مثل ما قلنا بنسميهم RAF MEK MAP kinase or هيدي فينا نسميها MAP kinase MAP kinase and then MAP kinase. Okay, that will lead to the changes at the transcription level mainly. Okay, I am done with this chapter. Do you have questions? No questions?
Doctor,
all is fine?
I have a question, please.
Yes.
Regarding type 1 TGF beta receptor هو كمان في عليه serine or threonine kinase domain, right? مش بس type 2.
Yes. Exactly.
Okay.
Exactly. The thing that you see in red, Samira, because
Yes.
also it has an enzymatic activity.
Okay. Regarding التايب 3 receptor tyrosine kinase coupled receptor ما نحن قلنا انه JAK family they are devoid of SH2 or SH3 so كيف بيعملوا binding على الستات 1 يلي في عندهم SH2 domain?
Let me just go to the type of receptor here. So قلنا نحن انه انت بدك تنتبهي لشغله سميره انه لحتى اثنين بروتين يعملوا انتر اكشن مش معناتها اثنينهم عندهم نفس الدومين. انا لما بقول انه بروتين عنده SH2 دومين عملت له definition لهيدا الدومين. قلت انا البروتين اللي عنده SH2 domain then this is a protein that has the ability to bind to phosphotyrosines يعني بدها بقبالها بروتين عندها تيروزين phosphorylated. لكن هون هيدا يلي عم نقوله. اوكي هلا شو بيصير عندك هون؟ انت ايه انا اللي عم اقوله انه الجاك والتيك يلي هن ار مثل علاقتهم بالريسبتور مثل علاقه الجي بروتين بالجي بروتين كابلد ريسبتور. This is a non-covalent interaction here. بس الترانس كونفورميشن تبع الريسبتور حتودي ليصير في عندي اكتيفيشن للجاك والتيك. سو انت لما بيتقربوا اثنين ريسبتورز من بعض بيصير في ترانس كونفورميشن، بتتغير الكونفورميشن a little bit. This will lead to هيدي الانزيم يلي هي هون جاك بتعمل فوسفوريليشن للثاني وهيديك بتعمل فوسفوريليشن لهي. Very good لحد هلا right. The next step is another phosphorylation that it will take place on tyrosines that are on the receptor itself. On الريسبتور في بقلبه tyrosine residues that will become phosphorylated. هالفوسفوتايروزينز هودي هن الدوكينج بوينتس لبروتين في بقلبها الاس اتش 2 يلي هي ستات. من خلال الاس اتش 2 الستات حتجي تعمل ريكوجنيشن تعمل بايندينج. ما لها علاقه للجاك، تعمل لها هلا الجاك تعمل لها فوسفوريليشن، بس ما بالضروره انه الجاك يكون عندها اس اتش 2 دومين لتعمل هالافكت وتعمل لها فوسفوريليشن للستات. اوكي يس الجاك والتيك كان المقصود منهم انه علاقتهم بالريسبتور منها من خلال اس اتش 2 دومين مثل علاقه جي ار بي 2 فور اكزامبل مع التيروزين كاينيز ريسبتور مع الانسولين ريسبتور. اوكي سميرة؟
Okay, doctor, thank you.
You are welcome. Other questions? If not, I will just finish the chapter here, and then next time we will start with chapter 2. You already have it, right, in the files?
About hypothalamus, right?
No need. Yes, the hypothalamus and pituitary. Both of them are
Yes.
going to be in the same chapter. Good. Have a good afternoon and see you on Wednesday.
يعطيك العافية دكتور.
Thank you.